Hello everyone, and welcome to Pearson Bio Academy. I am Fazna Imran.
Currently, we are focusing on Pearson Edexcel International Advanced Level, paper reference WBI 1201, Unit 2, which covers cells, development, biodiversity, and conservation. If you watched my introduction video, you know there are 6 key topics here. Today we are focusing on cell structure. I've broken it down into subcategories based on past paper questions. These include magnification, types of microscopes and staining, eukaryotic cells, prokaryotic cells, plant organelles, organization of cells, three-domain classification. Here you can see I've grouped the both plant organelles and three-domain classification under cell structure to keep things simple. Today, we'll be discussing questions on magnification from 2019 to 2024. To answer these questions, you need to know this equation from your book. Actual size is equals to image size divided by magnification. I've also made it easy to remember another formula. Magnification is equals to image size divided by actual size. In questions, they might ask you to find either the magnification or the actual size. Keep in mind to use the correct equation when you need to calculate one or the other. Before diving into these calculations, it's important to understand some basic unit conversions. For instance, did you know that 1 millimeter is equals to 1000 micrometers and it's equals to 1 million nanometers plus 1 centimeter is equal to 10 millimeter. Keeping these conversion in mind will really help as we move forward. If you have any questions or comments, please put them into the comment box. Now, let's move on to the first question from the June 2019 paper. In this question, you will see that they have provided an image of a sperm cell, and the question asks you to calculate the magnification of the sperm cell shown in the diagram. And this question is worth 2 marks out of 80. First, you need to get the scale measurements rather than the actual image length. In this case, the scale length is 4 cm, which is equivalent to 40 millimeters. You can measure this using a ruler. Remember, the measurement can be plus or minus 1 millimeter from 40. Now, with the scale length of 40 millimeters, convert it into micrometers. So, 40 millimeters equals 40,000 micrometers. The actual length of the scale bar is 60 micrometers. Next, we have to apply the equation for magnification. So, magnification is equals to image size divided by actual size. In this case, the magnification is 666.67. However, your answer can fall within the range of 666.67 to 683.3. Finally, when you're giving your magnification, don't forget to include the time symbol before the number. Now moving on to the second question. It's quite similar to the first one. This time, we are given a picture of a cell under a light microscope. You can see the image clearly here. The microscope has magnified the cell by 200 times, so the magnification is already provided. The question is asking us to calculate the actual width of this cell by measuring the line XY, and the final answer should be in micrometers. Let's break this down. Still, the question is worth 3 marks out of 80, so it's better to show a clear step-by-step process. First, just like we did before, we'll measure the length of XY using a ruler. In this case, it comes out to be around 26 mm. Remember, this value could vary slightly depending on your measurement, but let's go with 26 mm for now. Next, we need to be careful with how they've asked us to present the answers. Sometimes they may require the answer in standard form or rounded to 2 or 3 significant figures. So always it's better to read the question carefully. Now to convert the measured length into micrometers, we know that 1 millimeter is equal to 1000 micrometers. So 26 millimeter is equals to 26,000 micrometers. The equation we'll use to find the actual width of the cell is equals to image size divided by magnification. Here the image size is 26,000 micrometers and the magnification is 200 times. If we divide 26,000 by 200, we get an actual width of 130 micrometers. The next question is from the January 2020 paper. We are given a picture of a mitochondrion this time, and the student has already measured the length of the structure labeled U as 52 mm. Since the measurement is provided, there is no need for you to take any measurements yourself. The actual length of this structure is given as 3 micrometers. To solve this, we need to convert the measured length into micrometers to match the units. This question asks us to calculate the magnification of this electron microscope image, and this question is worth 2 marks out of 80. First, let's convert the units. The measured length is 52 mm, which is equal to 52,000 micrometers. The actual length is 3 micrometers. Now to find the magnification, we can use this basic formula here. So the magnification is equal to 52,000 micrometers divided by 3 micrometers, which gives us 17,333. Since we are not taking any measurement ourselves, there is only one correct final answer, and it is 17,333 times. And moving on to the next question is from the October 2020 paper. In this case, we are given a picture of a single cell called sailor's eyeball. The image is provided and the actual diameter of the cell is given as 25 micrometers. This question asks you to find the magnification, and you can see it's worth 2 marks out of 80. So what do you need to do? First, you need to draw a line across the diameter of the cell using a ruler. After that, measure the length of the diameter. In this case, the measured image length is around 42 millimeters, but it can vary slightly. Next, we need to do the conversion as usual. Since 1 millimeter equals 1000 micrometers, 42 millimeters equal 42,000 micrometers. The cell's actual diameter is already provided and it's 25 micrometers. Now we have to use the same equation, which gives magnification is equal to image size divided by actual size. So here magnification is 42,000 divided by 25, which gives a magnification of 1680 times. And remember to include times symbol to indicate the magnification factor. The next question is from the January 2021 paper, and it's about a mosquito. You are given a scale bar and told that the actual size of it is 4.4 millimeters. First, you will need to take the measurement. The image length is approximately 40 millimeter with a possible variation of plus or minus 1 millimeter. The questions ask you to calculate the magnification, and it specifically says that your final answer should be to 1 decimal place. Be sure to give the answer as it is asked. If not, you could lose your marks. And you can see this question is worth 1 mark out of 80. Now we have measured the image length as 40 millimeter, and the actual length is given as 4.4 millimeter. And since both measurements are in millimeters, there is no need for any unit conversions. The formula we'll use is the same. Magnification is equals to image size divided by actual size. And in this case, it's 40 divided by 4.4, which gives us 9.091. Since the questions require the answer to 1 decimal place, the correct final answer is 9.1 times. The next question is from the 2021 June paper, and it concerns a bacterium called Methanobrevibacter smithii, often abbreviated as M. smithii. You can notice that a scale is provided, and the questions ask you to find the magnification. And be sure to read the question carefully, as the answer should be presented in standard form to 2 significant figures. First, as usual, you will need to measure the length of the scale using a ruler. Here, the image length is around 35 millimeters with a possible variation of plus or minus 1 millimeter. Since the actual size is given in nanometers, we need to convert the image length into nanometers as well, to ensure both measurements are in the same unit. Therefore, 35 millimeter is equals to 35 million nanometers, and the actual length of the scale is given as 1200 nanometers. So we will use the same formula as before, and here we have to divide 35 million by 1200, which gives approximately 29,167. Since the question requires to the answer in standard form and 2 significant figures, we express this as 2.9 10 to the power 4 times of magnification. The next question is from the October 2021 paper. Here you are given a picture of a phloem vessel, and the magnification is provided as 200 times. The question asks you to find the width of the phloem vessel along the line XY in micrometers. Be sure to present your answer in micrometers and in standard form. To begin, you will measure the length of XY, which is about 38 millimeter here, with a possible variation of plus or minus 1 millimeter. Since we need to provide the answer in micrometers, we will convert the image length 38 millimeter into the micrometers. So here 38 millimeter is equals to 38,000 micrometers. And with the magnification known, we can find the actual width using the formula. The other formula you have to use here, actual width is equals to image size divided by magnification. And plugging in the values, we have actual width is equals to 38,000 micrometers divided by 200, and this calculation gives us an actual width of 190 micrometers. The next question is from the January 2022 paper focusing on Listeria monocytogenes. You are provided with a picture and asked to find the magnification. The actual length of the bacterium is given as 0.5 micrometers, and you'll use the label part X for your measurements. To begin, you should measure the length X with a ruler. Be very specific with your measurements as this length is quite small. For this question, it's 8 mm with a possible variation of plus or minus 1 mm. First, we need to convert the measured length into micrometers as usual, and here it's simple. 8 mm is equals to 8,000 micrometers. The actual length is given as 0.5 micrometers. Then we will use this value for our calculations. And we are using this same formula for this calculation. And here m is equals to 8,000 divided by 0.5. And the answer is magnification of 16,000 times. Since there are no specific requirements for how to present the answer, you can simply state it as 16,000 times. Additionally, based on paper variations, the final magnification could range from 14,000 times to 18,000 times depending on the student's measurement. The next question is from the June 2022 paper which focuses on the IVF procedure. You are provided with a picture of a fertilized egg cell and the magnification is given as 200 times. Your task is to explain how to determine the actual size based on information provided. Therefore, you don't need to take any measurements. It's essential to be clear in your explanation. You can state that to find the actual size, first you'll need to take the measurements of the egg cell. Once you have the image size, you calculate the actual size by using this formula, where actual size is equals to image size divided by magnification. Or simply you can say by dividing image size by magnification. And please note that if you provide any unmanipulated equations in your response, they may be ignored according to past paper marking schemes. Therefore, it's crucial to express the relationship clearly. The next question is from the January 2023 paper, which presents a photograph of a chloroplast. The question states that photograph B shows a chloroplast as seen using a different types of microscope, likely an electron microscope based on the details visible in the image. And your task is to find out the magnification. And it's important to provide your final answer in two significant figures. As usual, we'll start by measuring the scale provided in the image. In this case, the measurement is about 22 mm, which could have a possible variation of plus or minus 1 mm. Next, we need to convert this image length in micrometers, and here 22 mm is equal to 22,000 micrometers. The actual length of the scale bar is given as 1.5 micrometers, and to find out the magnification, we'll use the same formula. So, substituting in our values, we calculate m is equal to 22,000 divided by 1.5, which gives us 14,666.67. Since the question requires the final answer to be in two significant figures, We'll round this to 15,000 times. Thus your answer could be fall in between 14,000 to 16,000 times. Now let's move to the question from the May 2023 paper. It's about a sperm cell, which are quite similar to the question we encountered in the 2019 paper. The same picture of a sperm cell is provided with a scale, and you are asked to calculate the magnification. However, there is a slight difference this time. In the 2019 paper, you were only asked to find the magnification, but in this 2023 paper, you are specifically required to provide your answer in two significant figures. And this question is worth 2 marks. As usual, we'll start by measuring the length of the scale bar using a ruler. The measurement is about 4 centimeters, which is equal to 40 millimeters, with a possible variation of plus or minus 1 millimeter. Next, we'll convert the image length into micrometers. So it's 40,000 micrometers, and with the actual length of 60 micrometer, you have to use the same formula, just substitute, and the m is equals to 40,000 divided by 60, and it's 666.67. And since the question asks for the answer to be in two significant figures, we'll round it to a range of 630 to 670 times. As you can see, the question is similar to the one in 2019, and practicing these past paper questions really helps you to develop the skills needed to tackle future exams. This is the ultimate goal of our video series. And next, we have a question from the November 2023 paper, where you are given an image of pollen grains taken using an electron microscope. And the task is to find out the magnification, and the answer must be presented in two significant figures. As usual, the first step is to go for the measurement, to take the measure of the length of the scale bar, and here it's about 30 millimeter with a possible variation of plus or minus 1 millimeters. Now let's do the unit conversion. 30 millimeters is equal to 30,000 micrometers, and the actual length of the scale bar is given as 20 micrometers. So again, we have to use this formula, and here m is equal to 30,000 divided by 20, and it's about 1,500 times. So since the answer is already in two significant figures, there is no further rounding needed. However, if your measurement was 29 millimeters or 31 millimeters, you would adjust the answer accordingly, making sure to round it to significant figures. Finally, a last question from the January 2024 paper. The photograph provided shows some pollen tubes, and the stated magnification under the picture is 100 times. The task here is to explain how to calculate the actual length of the pollen tube. You don't need to take any measurements for these questions. So let's move on to the explanation. First, simply measure the image length of the pollen tube in the photograph. To find the actual size of the pollen tube, you would divide the image size by the magnification. You can also present this using the formula. And it's important to note that any unmanipulated equation should not be presented in your response. Therefore, you should avoid mentioning any alternative formula, such as the one that states Magnification is equals to image size divided by actual size, as this is not applicable to this question. Additionally, in the marking scheme, there is another answer that has been introduced. It states that you could calibrate an eyepiece reticule with a stage micrometer to calculate the actual size of the pollen tubes. This calibration process involves using the stage micrometer, which has a known scale, to ensure that the measurements you take with the eyepiece reticule are accurate. By doing so, you can obtain precise measurements of the pollen tube's actual length based on the calibrated scale. That's all for today's video. I hope you found this explanation helpful. If you did, make sure to hit the subscribe button and turn on notifications and you don't miss any future videos. Don't forget to like, comment, and share with your friends who might find this useful. Thanks for watching and see you in the next one. Bye bye.